Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our recent summer 2018 issue, and is titled Antibiotics and Antimicrobial Resistance, a Global Perspective. The article was written by Professor Anthony Coates, CSO of Helper B Therapeutics, and Dr. Yan-Min Hu, Senior Research Fellow at St George's University of London. So now on to the main article. Antibiotics and Antimicrobial Resistance, a Global Perspective. Antibiotic resistance is at a crossroad. In the world today, deaths in low-income countries from antimicrobial resistance, AMR, are predicted to rise dramatically by 2050. We know how to stop this disaster, and some steps are being taken in the right direction. But will these steps be enough? Antibiotics for critical priority bacterial pathogens are in the clinical trial stage, but, overall, the pipeline is described by the WHO as inadequate. Combinations of antibiotics and a rejuvenation of old antibiotics are appearing in development and have potential alongside new chemical entities and new classes. The AMR report, chaired by Jim O'Neill, has recommended what we need to do. The actions in response to this report are, at best, patchy. Universal access to new antibiotics by both the rich and the poor may be feasible, but universal stewardship will be a difficult problem to solve. Long-term solutions, particularly for antibiotic development by the pharmaceutical industry, are proposed in this episode. It is predicted that 10 million people will die each year from antimicrobial resistance if the present well-described inertia continues. These deaths will be predominantly in low-income countries. A limited number of brave, mostly small antibiotic companies are developing new antibiotics against critical priority pathogens, which are carbapenem-resistant gram-negative bacteria. But this alone is not enough. These new antibiotics need to become available to the poor in low-income countries. Furthermore, all countries need to use antibiotics in a responsible way, for example, not via the internet or over-the-counter, so-called stewardship, which will reduce the emergence of resistance. New, fast, cheap diagnostic tests will help this process when they become available in all countries. Since systemic antibiotics give rise to resistant bacterial mutants in the large intestine, AMR will continue to spread unless the use of antibiotics is substantially reduced, or some other solution is found. The review in this episode will discuss the WHO list of antibacterials in clinical trials from 2017, and we will then address the difficult issues of universal access, universal stewardship, and long-term antibiotic development by the pharmaceutical industry. Antibiotics which are in clinical development. WHO Antibacterial Agents in Clinical Development 2017. The World Health Organization, WHO, has identified 3 critical priority pathogens which threaten to significantly contribute to the increase in annual global deaths from 0.7 million now to 50 million by 2050. These are common bacteria which are highly resistant to antibiotics, including the most sophisticated penicillins, namely the carbapenems. The most dangerous of these are the carbapenem-resistant Enterobacteriaceae, because they are epidemic. This means that they spread rapidly with a doubling time of 2 years. They are also rapidly acquiring resistance to the last resort antibiotic, colistin. There are 6 antibiotics in clinical trial development which are active against all 3 WHO critical priority pathogens. Compounds in clinical development which are active against all 3 critical priority pathogens. Of these compounds, 2 have a new target. 5 were in Phase 1 and 1 in Phase 3, as reported by the WHO in 2017. Interestingly, 4 compounds were in combinations of which 2 use beta-lactamase inhibitor plus an old antibiotic. If the beta-lactamase inhibitor has only weak antibacterial action on its own, it is unlikely to reduce the emergence of mutant beta-lactamase, which results in resistance. Some beta-lactamase inhibitors are not active against all carbapenemases. Of the other two compounds, the mechanism of action of the synergy appears to involve increased membrane permeability. The likelihood of Phase 1 compounds reaching the market is 14%. The Phase 3 compound is more likely to reach the market. This pipeline is inadequate because it is uncertain that these compounds will eventually reach the market. Furthermore, according to the WHO, this pipeline will not be sufficient to tackle the impending AMR threat. Helper B Therapeutics has developed a new combination with azithromycin, a new class of antibacterial with a new target, and colistimethate sodium, colistin. The mechanism of action of azithromycin is to bind to the end of the replicating DNA strand, which blocks DNA strand elongation. Eventually, the block can be relieved by the cleavage of azithromycin from the strand, which allows elongation to resume. The combination is synergistic against colistin carbapenem-resistant CRE. The synergy opens up the development of low-dose colistin regimen. The combination is faster acting than colistin alone. It is also active against CRAB and CRPA. Because the two compounds are already in clinical use, the FDA and EMA allow facilitated clinical development paths. GMP supply is established, and most preclinical toxicity work has been completed, so the development time should be quicker than for NCEs. Importantly, the two compounds are both antibacterial. Therefore, the enhanced combination of the two should be able to reduce the emergence of resistance. Further compounds in clinical development which are active against one or two critical priority pathogens. There are further 12 compounds in clinical trials which are in this category. Of these, 7 are combinations, all of which are beta-lactamase inhibitors. Clinical trial stage compounds, 3 are in Phase 3, 2 are in Phase 2, and 6 in Phase 1, and information about one is not available. These compounds will be particularly useful in situations where the susceptibility of the pathogen is already known. For example, after conventional or rapid diagnostic tests. Where the pathogen is unknown, these new compounds could be useful in combination with other antibiotics if it is suspected that the spectrum needs to be broadened. The case for the development of combinations of antibiotics. Rejuvenation with combinations and reduction of resistance emergence. Combinations of antibiotics are used to rejuvenate old antibiotics. The classic example is the combination of beta-lactam and a beta-lactamase inhibitor, such as amoxicillin plus clavulanic acid. Of the 18 compounds in clinical development, with activity against one or more critical priority pathogens, half are combinations with beta-lactamase inhibitors. Companies such as Spero and Helper B Therapeutics are developing non-beta-lactamase combinations which rejuvenate old antibiotics against all of the WHO critical priority pathogens by permeabilizing the bacterial membrane. Another important aspect of combinations is to prevent the emergence of bacterial resistance to antibiotics. In 1948, streptomycin was used to treat tuberculosis patients. Although improvement occurred initially, resistance developed and many died. In 1952, the British Medical Research Council completed a landmark trial in which they treated tuberculosis patients with a combination of streptomycin and para-aminosalicylic acid. In this case, resistance did not develop and all the patients survived. This showed that combinations can prevent the emergence of resistance in patients with tuberculosis. This finding has led to the development of the standard 4-drug combination regimen, which is used to treat tuberculosis today. Unfortunately, if this standard TB regimen has not been used properly, for example, patients fail to follow the course of medication, resistance emerges. The way that combinations prevent the emergence of resistance is thought to be through the mutation frequency of the individual drugs in the combination. For instance, if we consider the combination of old antibiotics, A and B, which act on different targets, if A has a mutation frequency of 10 to 6 and B also has a mutation frequency of 10 to 6. The mutation frequency of the combination should be 10 to 12. In other words, a combination of the two drugs is much less likely to lead to the emergence of resistance than one drug which is used on its own. Combinations have also been developed for infections due to Helicobacter pylori and human immunodeficiency virus. These regimens also prevent the emergence of resistance. However, combinations have not been well explored for common gram-positive and gram-negative bacterial infections. Here, we describe a small number of examples. While a combination of trimethoprim and sulfamethoxazole was developed, resistance did develop, probably because the drugs targeted different enzymes in the same metabolic pathway. Combinations of beta-lactam antibiotics and beta-lactamase inhibitors are widely used, but resistance has also arisen to these, possibly because most beta-lactamase inhibitors are not themselves antibacterial, and so the accompanying beta-lactam antibiotic is, in effect, monotherapy, which quickly leads to resistance. In the author's view, the way forward is to re-explore the combination of two or more antibacterial agents, each of which acts on a different mechanism against the target bacterium. In the WHO list of antibiotics which are active against critical priority pathogens, Helper B Therapeutics is developing a Phase 2 ready combination of azithromycin and colistin. Both of these compounds are active against Enterobacteriaceae. Azithromycin is a bacterial DNA chain replication inhibitor and is a new class of antibacterial compound. The review on antimicrobial resistance, chaired by Jim O'Neill and published in 2016. This review recommended 10 interventions, sometimes called the 10 Commandments. 1. A massive global public awareness campaign. There has been some progress in some countries, but much more needs to be done. 2. Improve hygiene and prevent the spread of infection. There is already a wide range in standards between different countries. Although some new efforts are underway, there is a long way to go. For example, the spread of colistin resistance seems to be faster than expected, particularly in bacteria which are also resistant to carbapenems. 3. Reduce the unnecessary use of antimicrobials in agriculture and their dissemination into the environment. Some good progress in some countries, but still a long way to go. 4. Improve the global surveillance of drug resistance in humans and animals. There has been some progress in some countries. 5. Promote new, rapid diagnostics to cut unnecessary use of antibiotics. Significant progress has been made in the development of new fast diagnostic tests for antibiotic-resistant bacteria. Some of these tests are already in the market, and improved, even faster tests are in development. The AMR report suggested that rapid diagnostic tests should be a prerequisite for all antibiotic prescribing, but no government is enthusiastic about this idea. But for new diagnostics to reduce unnecessary antibiotic usage and provide effective therapy to those most in need, they must be embedded in clinics throughout the world. 6. Promote the development and use of vaccines and alternatives. Some progress is being made, but the absence of enough interest by big pharmaceutical companies and the lack of market entry rewards will slow this important area. 7. Improve the numbers, pay, and recognition of people working in infectious disease. Following the increase in AMR publicity and financing, the number of researchers in the area seem to be increasing. 8. Establish a global innovation fund for early-stage and non-commercial research. The news is encouraging, because the financing for early-stage research and development is increasing. For example, the Chinese and British governments, the Wellcome Trust, and the US Biomedical Advanced Research and Development Authority, BARDA, and Danish pharma company Novo, have announced significant new joint funding initiatives. 9. Better incentives to promote investment for new drugs and improve existing ones. Although representatives of the 25 largest pharmaceutical companies are thought to have discussed AMR at Davos 2018, they agreed to remain engaged on the issue. This is very disappointing. For example, serious interest in AMR by Big Pharma and market entry rewards as suggested by the O'Neill report, would stimulate and facilitate new drugs, vaccines, and diagnostic tests. And 10. Build a global coalition for real action via the G20 and the UN. Progress is slow. On the one hand, governments tend to move slowly. On the other, if all the people who die from AMR in 1 year were to die in 1 day, for example in plane crashes or fires, governments would move much faster than they have done over the past 2 years. Overall, progress during the past 2 years has been patchy. Some areas, such as the return of big pharmaceutical companies and the introduction of market entry rewards, have not occurred. This means that new drugs, vaccines, and diagnostic tests will struggle in the development and marketing stages. In the author's view, at the present slow rate of progress, the O'Neill projections for 10 million deaths per year by 2050 may be an underestimate. Universal access. Supply to the rich and to the poor. Antibiotics are life-saving drugs. Antimicrobial resistance means that many antibiotics are no longer effective amongst the poor. New antibiotics are going to be discovered and developed, mainly in high-income countries such as the US. But most of the burden of AMR disease is already falling upon lower-income countries and will continue to do so. In the 1980s, new drugs for AIDS were developed in high-income countries but were too expensive for the poor. Indian companies such as Cipla, led by Yusuf Hamid, produced AIDS drugs which were affordable by the poor for a fraction of the cost. This is a somewhat extreme commercial model. In the AMR field, there is a not-for-profit model called the Global Antibiotic Resistance and Development Partnership, GARDP. WHO was the midwife, DNDi the mother, and GARDP the baby, born in 2016. It has a remit to ensuring universal access. In addition, GARDP aims to develop antibiotics and antibiotic combinations which are not commercially viable, such as treatments for sepsis in babies and children, and for the highly resistant gonococcal infection. This model is not suitable for commercial development, although GARDP does partner with companies. An example of a commercial model, there are many, is Helperby Therapeutics, which has a partner in India which co-develops combinations of antibiotics and supplies to India and Africa at prices which are affordable by the poor, while Helperby sells into high-income countries. Stewardship. Antibiotic stewardship is coordinated intervention which reduces inappropriate use of antibiotics by helping the selection of the optimal antibiotic regimen, the dose, duration, and route. The global reduction of inappropriate antibiotic use is important because the administration of an oral antibiotic to humans results in antibiotic resistance in the large intestine within 7 days. The length of time that the resistant bacteria are detectable in the individual is up to 1 year. This means that each individual who has been treated with an oral antibiotic is defecating resistant bacteria for up to a year. Since many people in low-income countries do not have access to toilets, contamination of the environment with highly resistant bacteria occurs. It has even been recorded that up to half of visitors from a low-level resistant country who spend time in a country with a high level of antibiotic resistance return home carrying highly resistant bacteria in their large intestine. In high-income countries, where water-flushing toilets are usually available, the antibiotic-treated individual carries resistant bacteria in the intestine, may transmit them to their household, and may be a risk to themselves, for example, if they require surgery or become immunosuppressed. Furthermore, sewage systems sometimes break down and release raw effluent into river systems, thus contaminating the environment. Stewardship requires local guidelines and teams of trained professionals. In the UK, for patients with life-threatening infections, prompt, effective antibiotic treatment must be started within 1 hour, or as soon as possible. Broad-spectrum antibiotics should be avoided, and careful documentation should be recorded in the patient's clinical records. For prophylaxis, a single dose is recommended, or more doses in special circumstances. Once susceptibility is known, switch to the correct antibiotic. Ultra-rapid antibiotic sensitivity tests, which give results in less than 1 hour, would be helpful. national and local stewardship guidelines and regular reviews will be needed. Education of patients and their clinical and non-clinical caregivers should be a priority in the fight against AMR. The United Nations has recommended that every country plays its part in the control of antimicrobial resistance, and improved stewardship is an important component of these efforts. A global stewardship regulatory authority is a logical next step. However, at the present time, it is unlikely to be a practical solution. For example, internet purchase of antibiotics, particularly by people with poor access to these drugs, sale of antibiotics over the counter, provision of antibiotics by people who are not medically qualified, poor sanitation, poor education, war, and inadequate infrastructure are significant hurdles. Whether countries will adopt effective stewardship schemes by 2050 is an open question. In the first 2 years since the UN resolution, there has not been an obvious slowing of, for example, the epidemic spread of colistin resistance. This does not bode well for the global control of the spread of antimicrobial resistance. What should the pharmaceutical industry do to tackle the AMR problem? It is easy to blame the large pharmaceutical companies for failing to develop new antibiotics. However, it costs huge sums of money to discover, develop, and then market an antibiotic. It is well known that the unit price for a course of treatment for a life-threatening infection with a new antibiotic is lower, from 10 to 1,000-fold, than other new drugs, such as new cancer or immunologic treatments. Big pharmaceutical companies are not charities and answer to shareholders who require profits. What is clear now is that they will not come to the rescue, with a small number of exceptions, unless the financial rewards are realistic. What they should do, in the author's view, is to make a joint effort to persuade governments to introduce market entry rewards. A prize of $1 billion or more, as suggested by Jim O'Neill's AMR report, for the development of new treatments to combat AMR would make a significant difference. Nevertheless, the present situation is quite positive. In spite of all the challenges, a handful of mainly small companies have antibiotic treatments in clinical trials against all 3 of the WHO critical priority pathogens. A further 12 companies have compounds which are active against 1 or 2 of these dangerous pathogens. There are increased levels of grants and fast-track regulatory support. The first global public-private partnership has been founded and has raised substantial sums of money for the development of antibiotic treatments for infections which the commercial sector is unlikely to cover. New antibiotics which are effective against the critical pathogens will reach the market. How the market will respond is not clear. The optimistic view is that several of these will be blockbusters like Cubicin. In other words, will earn $1 billion or more per year. The pessimistic view is that they will struggle because it is fashionable to pay a low price for antibiotics. It is the author's view that some of these drugs will attain universal access for the poor, particularly for companies which have partners in low-income countries. Whether these new antibiotics will be used in accordance with good stewardship is unknown at present time. What is the 100-year plan for a pharmaceutical industry which faces the complete destruction of the current repertoire of available antibiotics? The simple answer is that there is no plan. Unfortunately, bacteria do have a plan. This is to become resistant as quickly as possible to all new antibiotic threats. It has served them well for billions of years. The author's view is that industry should have a long-term plan. The plan must be to find treatments for AMR as quickly as bacteria do. For example, to focus on developing new monotherapies for AMR and then throwing them away is unsustainable for 100 years. The plan should include old antibiotics. The rejuvenated old antibiotic would only be used in combination with another antibiotic in order to reduce the emergence of resistance. Combinations could be 2, 3, or even 4 drugs, as is the case in tuberculosis therapy. Ultra-fast diagnostics would be used to choose the correct combination. The current regulatory framework is much too slow to deal with AMR, which can arise in a few days. In order to populate a new range of antibiotic combinations of old and new antibiotics, ultra-fast clinical trials are needed. For example, Phase 1-2 PK/PD with less patients than at present. In this way, it could be envisaged that a single old antibiotic could be rejuvenated several times in a 100-year period by combination with different antibiotics, sometimes called antibiotic resistance breakers. That half of the 18 drugs in clinical development against critical priority pathogens are combinations with beta-lactamase inhibitors supports the idea that rejuvenation of old drugs is feasible in the long term. Conclusion: Antibiotics and Antimicrobial Resistance, a global perspective. Antibiotic discovery and development for resistant gram-positive bacteria has been successful. For critical priority carbapenem-resistant gram-negatives, the pipeline is inadequate, although the prospects are reasonably good for some drugs, even if they do not cover all of the problem bacteria. This article was written by Professor Anthony Coates and Dr. Yan-Min Hu. Professor Anthony Coates is the CSO of Helper B Therapeutics, leader of research teams, author of about 150 publications, recipient of numerous national and international grants from European Commission, British MRC, charities and industry, and named inventor of many patents. He is also the Professor of Medical Microbiology at St George's University in London. Dr. Yan-Min Hu obtained her PhD degree in the laboratory of Professor Coates in 1999. She started her postdoctoral research with Professor Coates in St George's Hospital Medical School, London. Now, she is a Senior Research Fellow in St George's University of London. Her scientific profiles are demonstrated by a stream of high-profile publications. If you've enjoyed this episode, then you can subscribe to the journal free of charge by visiting ddw-online.com, where you'll also be able to view full articles including references and download the original PDFs of past articles, including this one. You can find all the links in the show notes. If you've enjoyed the podcast, we massively appreciate you leaving us a review and subscribing, and you can also follow us on LinkedIn and Twitter. Thanks for listening, and we'll hope to see you in our next episode.